Hello,
Hello.
welcome. Let us know where you're joining from. Hello, สวัสดีครับ
Hello.
สวัสดีค่ะ
สวัสดีครับ Are you joining from กำเนิดวิทย์?
ใช่ค่ะขอบคุณนะคะ
That's very nice.
Hello, welcome. Thanks for coming.
Maybe you can talk about what you like about neuroscience.
Yeah, so when I was in undergrad, I was very interested in neuroscience because, the brain is so complex, as you can see in these two images. It's so beautiful. And I always wanted to see how studying the brain can understand why we behave a certain way, or if something goes wrong in the brain, how does it cause us some kind of disease, right? So I think that's why it's interesting. What about you?
Like most people, I think it's a pretty wild thing to see that this brain right here can, with all its connections and all its chemicals and electricity, can do something crazy like consciousness or memory or learn. And I just find that something as abstract and complex as those, when you can map it down to a bunch of connections, is something that still blows my mind till date. And it's just incredible how this nature made it this way.
I agree.
And I find flies very cute.
Yes, we'll be talking about different species from worms to flies to mouse to humans. They'll
Mhm.
mostly focus on flies and humans, but as you will see in the presentation, it wouldn't matter because in the end it's the brain. And the reason why we're studying these different models organisms is because it allows us to understand something better about cognition and diseases related to the brain.
Yeah. And even like you see a fly and a worm and you think, oh, well, it can't do much. It's not a human. It's pretty remarkable you find the stuff they can do, but we take for granted.
Yes. Do you know if anyone would be joining? If you're from KVIT? Okay. See someone.
I'm not sure, but the teacher sent me the link in the group chat, so I got the link and joined.
Okay,
I guess there'll be more more friends joining.
that's okay. Okay, so I think we'll get started. What's the time in Thailand? It's 10:30, I guess. It's 4:30 a.m. here in the UK, and for you, Arushi, what's the time?
It's 11:30 p.m.
Yeah, we heard you guys like Connectomics, and we wanted to talk about how cool Connectomics is and how it's quite essential to neuroscience. But anyways, to introduce myself, I'm Saponnat, that's in Thai, and I'm a PhD student at University of Oxford and the co-chair of the Fly Connectome 2025 Fly Connectome Research Competition or FlyCom 2025. Or if you can just admit people in, that would be great. And I personally use computational neuroscience to model the brain using the human connectome for my PhD, and I kind of did that in undergraduate as well. And today I will be co-leading this workshop with Arushi Vardhan, who, as you can see on the screen, Arushi is also our FlyCom 2025 co-chair and research lead. She's the academic ambassador of biological sciences at Clematis. She is also a graduate student in the Department of Cell and Systems Biology at University of Toronto. Her research focuses on the connectome-driven exploration of neuroconnectivity in the Drosophila visual system. And today we have actually, very exciting stuff for you guys. But before we start, I want to thank you all for being a part of the Connectomics journey so far. If you are new here, we hope this opens some new perspectives to what we can do in neuroscience. Also, before we start, I also want to thank a few people. Special thanks to Om, who I've met at Oxford, and he helped us outreach to students at Connectwit or KVIT, which is a high school in Thailand. Also, thank you to KVIT teachers and students for being here. And I hope for those who can't be here, you guys can watch this recording. It will be recorded and uploaded on YouTube. I also want to thank Ali from the University of Tripoli in Libya for reaching out to Libyan medical students. And thanks to Aditya, as well as Sohaib, Sean, Kauzier, our FlyCom 2025 ambassador, for reposting our post on LinkedIn. So that's my thanks to all the people who have helped make this possible. And now let's go to what we're actually going to talk about today. So the first thing we're going to talk about is what really is connectomics. I'm sure you might have heard this in the news, but I want to really spend the time with Arushi on what exactly is connectomics. And we will also give an overview of the competition itself. We will then talk about different types of connectomes or connectomics. We will then discuss the work that our guest speakers for FlyCom 2025 have been doing, and a lot of these work really shows the recent breakthroughs in fly neuroscience and in synapse-level connectomics. We will also go over some common feedback and tips for the competition, and lastly, we will open it up for questions. Though a lot of the stuff we'll be talking about is quite complex. You can always talk in the microphone to interrupt us at any point in time. So what is connectomics? So connectomics is the production and study of connectomes. And what's the connectome? Connectome is brain maps. But that's it. If it's just about the brain map, then why is it so important to neuroscience? Like, why should we care about brain maps? Is it really critical to in the field of neuroscience? And the answer is yes. In fact, we need to understand brain maps to do most of the things in neuroscience. And it's kind of encapsulated in this quote. So in this quote, it says, if we want to understand who we are and how we think, part of that is understanding the mechanism of thought. And the key to that is knowing how neurons connect with each other. In other words, we need to have brain maps to really understand the neuromechanism of cognition and also what goes wrong in that thought, right? So what exactly are brain maps? So when you think of brain maps, there are two things to consider. Or in other words, if you think of connectomics, there are two kind of categories that we should be thinking of. So first is the scale. In other words, the scale is the level of detail that this map is giving. So is this map giving us the level of detail to the level of synapses? In other words, can we actually look at the connections between two neurons or two cells? So that's the synapse level kind of detail. Or is it more macro scale, which is macro is anything that's not at a synaptic level? Then we have the second category, which is this map a structural map or is this map a functional map? In a structural map, you basically see the neuroanatomy of the map. So you see, oh, how do these two neurons look? How do these two neurons connect to each other? The functional maps, on the other hand, is a map that shows how different areas have different kind of activities that are going on. So a functional map records brain activity. To get a sense of different kind of connectomes, let's try and do a short quiz. I encourage you to try shout in the mic or otherwise you can type in the chat. Let's say we take a brain scan of a human brain. So let's say I go on an MRI scanner, and you might have heard of MRI. They are always used in hospitals, and you basically take a brain scan. The question is, do you think brain scan like this is a structural map or a functional map? If you guys could type in the chat, if you would like to engage via microphones, no, that's fine. Okay, someone says structure. Someone has DM'd me. Yes, MRI scan is a structural scan or a structural map. Do you think an MRI scan is a microscale or a macroscale kind of map? So remember, microscale is when you look at individual neurons, and macroscale is kind of anything bigger than that. So when you take an MRI scan, can you look at the level of neurons? Yes, two people answered it's a macroscale. That's great. Great job. Okay, this is a very beautiful image from one of the papers. And this is basically a whole brain imaging or whole brain recording of the brain activities in zebrafish. And they kind of use light sheet microscopy to do it. Do you think this kind of map is a structural map or a functional map? Yes, that's great. We have some responses. So functional map. And as you can see, these images look really, really cool. So it is with a zebrafish. But essentially you can measure the activity of the brain in humans too, which we'll see how we can do it. So we can do it with flies, we can do it with humans, we can do it with zebrafish. Okay, do you think this map, this functional map, is at a microscale level or a macroscale level? So remember, at a microscale, you're looking at the level of neurons or level of individual neurons. And macroscale is anything bigger than that. So we have some mixed responses. So I'll give you a hint. So obviously it's not clear from the image, but look at the caption of this figure. So you're using light sheet microscopy. So actually, this is very cool. But when they did this functional recording, they were recording at the level of single neuron, and they were able to do it with zebrafish. So now, do you think that's macro or micro? Great. So it's actually microscale. Just to reiterate what structural and functional is. So when we are recording a neuron's activity, we call it a functional map or functional imaging. So functional refers to some kind of activity in the brain, and structural refers to the neuroanatomy of the brain, right? I hope we make those distinctions clear. So now hopefully that you understand the difference between structure and function and microscale and macroscale, we can understand now what we're going to talk about in this workshop today. So first we will talk about the microscale connectome. Then we'll talk about macroscale connectomes and functional connectomes. And the macroscale will kind of focus on humans. And then also in humans, we'll kind of focus on brain pathology and how connectomics is essential to understand some of the brain pathology in humans. Then we will talk about how synapse-level connectomes can help us understand a bunch of things in cognition and whether spending huge amount of resources in building a very detailed neuronal wiring diagram are they really useful. Then we will also show how you can be involved in this connectomics analysis, and we'll use two tools, Neuroglancer and neuPrint. And this kind of analysis will be very relevant for our competition as well. And we will also kind of go over on some tips and tricks and some feedback based on the submissions that we have been seeing. Okay, so just a very quick overview for those who are new to FlyCom 2025. But essentially, very recently, scientists have created one of the most comprehensive wiring diagrams of the brain, and we're talking about the microscale one, right? It's a synapse-level connectome. So in other words, the study of how neurons connect to each other, we call this connectomics, as we know, and we'll be using this very fascinating data set that was just released a few months ago to uncover new insights about the brain. This data set, which we call the optical lobe, has over 49 million synapses and 53,000 neurons, so you can imagine how massive this data set is. And together, Arushi and I, and on behalf of our faculty partner, Professor Eyal Grutman and Inan Ryder at University of Toronto, want everyone here to be a part of our learning journey to use powerful connectomic tools to visualize and analyze the connectomics dataset. And by studying neuronal connectivity, you can learn so much about the brain. And I might have used this analogy in the previous workshop, but it's kind of like diving into a vast ocean to find how thousands of species interact with each other. So today we will also demonstrate how to use these cutting-edge connectomic tools to learn something new. So one of the tools is called Neuroglancer. So Neuroglancer is used to visualize a lot of these synapse-level connectomics data. And the synapse-level connectomic data is also volumetric because the brain is in a 3D space. We will also be demonstrating how to use neuPrint, which is to query neuronal connectivity, or kind of how two neurons are structurally connected with each other. So hopefully you guys will find this exciting. And the challenge for the competition is that you'll be choosing one neuron. Remember, there are 53,000 neurons. So you'll choose one neuron using the Neuroglancer, which is the visualization tools, or you can also choose a neuron from the literature. Once you choose a neuron, you will kind of try to explore the structure of this neuron. Remember, a lot of times our brain or our neuron is shaped a certain way because it helps us with some kind of function. Otherwise, why would our brain evolve to have the structure that we see today? The third thing is to analyze the neuronal connectivity using neuPrint. And the fourth step, and the very important step, is to propose how your neuron processes information and contributes to fly behavior. And essentially, we'll be combining the connectomic analysis using these two tools and look at the literature, kind of integrate this into two deliverables. One is the creative piece. So creative piece is like a poster, an art, infographic, whatever you may like. But essentially, you would like to ideally communicate about your neuron and about how you have done such a great job, explaining the structure and the function and your proposal, but you want to communicate this to a public audience so that if someone who do not know anything about fly neuroscience, about neuron, look at your submission and be like, wow, this neuron is so interesting. And the second deliverable would be a written research report, which is 500 to 1,000 words. And there are some important deadlines. So the competition runs from May to December. The early feedback opportunity deadline has just passed. And for those who submitted before August 31st, we'll be giving feedback by sometime early October. We have the final submission deadline on December 1st, and the winners will be announced in January 2026. And remember, you will also be competing for 500 Canadian dollars in total prizes, and we have two separate categories, high school and undergraduate. So now I will let Arushi talk about the Synapse-Level connectomes, and meanwhile, I will send folks some links to the competition website so you can have a look at it after the workshop. Go ahead, Arushi.
Hey, all right. So I will jump into a brief history behind synapse-level connectomes. So to start off with, what are even synapse-level connectomes? You and I and most animals have neurons that connect to each other, right, in the brain. And the space between them is called the synapse. Now, synapse-level connectomes really kicked off around in the 1980s with the release of a whole brain connectome of a worm called C. elegans, which, as you can see here, it's a tiny little worm. And it had all the neuronal connections of this worm. although this is a tiny worm with around 300 neurons, the task to actually even get there was a very, very challenging one. They had to take this worm, they had to slice through it and image it using electron microscopy, as you can see here. This is one slice, and actually trace through that neuron's journey through each of these slices to reconstruct these neurons. So it's a completely human-made endeavor without much technology to support. But cut through 2024. Now we have a full brain fly connectome with around 140,000 neurons and 50 million chemical synapses. And the reason we could even get here was because of the recent advances and breakthroughs in technology and AI, in imaging, and other things that could make it possible to even have a full brain fly connectome, and one of which you are using in the fly connectome competition. And this is the age of many synapse-level connectomes emerging. Another example is of the Microns project. This project mapped 1 millimeter cubed of the mouse visual cortex. And the cool thing about this connectome is that it's not just an anatomical connectome, but it also has functional information within it as well. And similarly, we also have a millimeter cube of a human cerebral cortex, as you can see here. So yeah, these are the ages of synapse-level connectomes, but just to keep in mind, a millimeter cube is like the size of a sand. So, and already a size of sand, as you can see, is like 1.4 petabytes of EM data. So there's still a long way to go, but the future looks exciting for Connectomics. Yeah, and I'll give it back to Sapo.
Thank you. Yes, and remember these synapse-level connectomes, they are so hard to reconstruct. And right now, the current breakthroughs, despite the advancements in AI and image reconstruction and so on, we are only able to get 1 millimeter cube of the brain, to actually get a complete synapse-level connectome. And as you can see, it's so hard to do a synapse-level connectome. But why do we need Synapse-Level Connectome? Well, it's because we can see everything in such great details. So this is an example of Neuroglancer neuPrint of the data set that we'll be using. So I will be demonstrating this live, but I'm here just to give you a teaser of what you can do with Neuroglancer. So in Neuroglancer, once you click on the platform, you can actually look at different neuron types. And remember, there are around 50,000-ish neurons, and there are different neuron types. Neuron type is basically the type of neuron. So although there are 50,000 neurons, there are around 730 neuron types. So one neuron types, there could be, let's say, 500 neurons that belong to the same category or same neuron types. Just like when you go to a supermarket, you might see chips, right? You might see Lay's, you might see Cheetos, you might see a bunch of chips. But for Lay's, you might see 10 green Lay's. So that green Lay's is like a neuron types. You can also look at whether this particular neuron type has which kind of neurotransmitter. And remember, neurotransmitter is the chemical messenger that sends and that binds to the synapses. And neurotransmitter can influence the activity of the neuron. It could be inhibitory, which means it could reduce or suppress the activity of that neuron. It could be excitatory, which means it could increase the activity of that neuron or increase the probability that neuron will fire. Of course, because it's a synapse-level connectome, you can see synaptic sites. So you can see where in this particular neuron, is it receiving information, is it receiving incoming information, or it's giving away information. So post-syn stands for postsynapse, and pre-syn stands for presynapse or presynaptic sites. With neuPrint on the right side, let's say we'd have C2, right? Now you can actually look at how C2 connects to other neurons. So I don't know if you can see, it's a bit small here, but this circular chart essentially tells you which particular neuron or region is this receiving inputs from and sending outputs to. So essentially you can use neuPrint to analyze neuronal connectivity for that particular neuron of interest. And this is why it's so cool to use a synapse-level connectome, because of the detail that you can get from this. Before we talk about synapse-level connectome, I kind of want to talk a bit on the macro scale. And keep in mind that even when I talk about the macro scale connectome, the same principles applies to all sorts of connectome. So what does it take to build a connectome? I think one of the three ingredients is that first you kind of need a structural map with brain regions, with the boundaries of brain region clearly marked. And a simple analogy is, let's say we have a map of Bangkok, which is, the capital city of Thailand, and you have, let's say, 50 districts. Now, how do I know which house belongs to which district? In order to do this accurately, you need to have a clear boundary as to, like, this particular district where does the boundary lie? So we need this kind of structural map. So that's the first thing we need. The second ingredient is we need a structural connectivity map showing fiber tracks that connect different areas or that connect different neighborhoods. In other words, we kind of need, so instead of having just a map like this, like in number one, so instead of that, now you add roads to show connections between houses, to show connections between restaurants, or to show connections between districts, like a highway perhaps. The second ingredient is to create a structural connectivity map. Now, the third is to create a functional connectivity map. It basically shows which areas are active at the same time. So remember, function refers to the brain activity. And an example is, how do we know if the information is flowing from one area to the other? In other words, if you think of a city map analogy, it could be a traffic kind of map. So you can look, the rows in red means there are a lot of cars. And this map is also time dependent. It's possible that during peak hours, there are a lot of cars. The same thing with our brain. When we're trying to, let's say, memorize something, there might be a strong functional connectivity between area related to memory and maybe area related to vision because you need to see things in order to memorize things. So in other words, if two areas are active at the same time, we say there's some kind of functional connectivity with each other. And just a note that sometimes two brain areas that have no physical connections can still have similar brain activity patterns. In other words, two brain areas do not have to be physically connected to each other. Even if that's the case, they can still have strong functional connectivity. So just keep in mind that functional connectivity doesn't always depend on structural connectivity, though oftentimes it does. Now that we kind of understand that analogy, let's go step by step of how we can build macroscale structural and functional connectome. And I know this graph might look a bit complex, but we'll go one by one. So the first step to building structural connectome, and actually the functional connectome as well, is to create an atlas where you can find boundaries between regions of interest that we're interested in. So how do we do this? So again, the analogy is we are trying to find boundaries between each district in a city or each city in a country. So the way you do that is first you take a structural MRI scan. So just like how you would go to a hospital and you might get an image like this and your doctor might be like, oh, I can see there's a tumor or there's something wrong with the brain, something wrong structurally with the brain. So that's the same MRI scan I'm talking about. Once you take the structural scan, you now try to label the boundaries of each region. Okay, so for instance, you can take that brain, you can overlay it with some kind of atlas, which explains like, okay, this particular region's like frontal pole has this particular landmark and the boundary should be divided here for this particular person's brain. So essentially, it's important to label the boundaries of each region. And when we mark the boundaries, we call this process parcellation. So if you go read a lot of Connectomics papers, you will see, oh, we have done XYZ kind of parcellation. So that's what this means. And depending on the type of analysis you want to do, you can have different kind of parcellation. So you can have, let's say, a higher resolution parcellation on the left side of the screen. So as you can see, the brain areas are divided more fine-grained. And on the right, we have like a lower resolution parcellation where the atlas kind of make the boundaries like a bit bigger. So it really depends on the type of analysis you want to do. What's the point of taking a structural map with the brain region boundaries clearly marked? So an example, if you take an analogy of the city map is how large is a certain district or how large is Lat Krabang, let's say, which is one of the districts in Bangkok. And the only way you can do this if you know the boundary of the city or of the district that you're interested in, then you would kind of measure the area, right? So same thing for the brain. Because the brain is in 3D, it's a volumetric data set, you can basically measure the volume of the brain and you can correlate it with some kind of metric in cognition. An example research question that you might be thinking of is, does hippocampal volume relate to cognition among older adults? So hippocampus is often, it's a structure related to memory. And of course, very important cognition, because if you don't memorize, if you can't remember things, then you basically have cognitive impairments. So for instance, this particular paper that I've screenshotted is trying to see if hippocampal volume relates to cognition in some ways. But you can also study because you can delineate hippocampus from both the sides. You can see if, like this paper I also studied, if the asymmetry between, or if the differences in the hippocampal volume on the left versus on the right side, does that influence the cognition in some ways? So you can do all sorts of things if you can have a clear boundary of a certain region. Another example is you can use the structural metrics to predict some kind of treatment outcomes, right? So an example here is, can we use a volume in hippocampus to predict whether a certain treatment, like ketamine, which is a drug, so can we use hippocampal volume to predict whether ketamine would be successful in helping some kind of disorder, in some kind of treatment? So in this case is helping people with suicidal thoughts. So just to reread, hippocampal subfield volumes, or volumes in certain sub-areas of hippocampus, does it predict treatment response or does it predict treatment outcomes to oral ketamine, which is a kind of drug, in people with suicidality? So essentially, that's what that research is trying to say. And just another note, hippocampal volume here is basically a biomarker or a biological indicator for some kind of treatment response. And essentially, these kind of metrics, these neuroimaging measures can be used as a biomarker for many things, for health, for disorders, for predicting treatment outcomes, or for treatment selections, and so on. The second ingredient is structural connectivity, right? You now want to show the connectivity between two regions. How do you do it? So you basically take diffusion tensor imaging scan. Again, it's a type of MRI. Essentially, the TI allows you to reconstruct the images of axons or fibers or white matter tracts in the brain that connect between brain regions. And as you can see the image on the left, you can see the DTA allows you to actually visualize the tracks or the fibers, the neuronal fibers between different regions. And how does it do it? Again, DTI is actually, you use the same machine as the MRI, by the way. So how it works is you use magnetic field to measure the diffusion of water molecules. So you can imagine that if you have a long axon or a long neuronal fiber, it kind of makes sense that the water diffuses faster along this direction of axon, but it diffuses slower in this direction. As you can see the arrow that says slower diffusion, and that kind of makes sense because in the direction where it says slower diffusion, there's a lot of cell membrane which doesn't allow water to diffuse at all. And by capturing this kind of water diffusion, you can infer how the track looks. That's why we call it reconstructing the images of the fibers. So you're not directly imaging the fibers, you are measuring the water diffusion and you're reconstructing the image out of it. Now you can see how the tracks connect between two regions. We call this a structural connectivity. And of course, a lot of things influences these density of white matter tracks. So you could imagine that if you age, the white matter tracks kind of gets less dense, right? Which is very intuitive. Again, we can use measures like DTI or structural MRI as a biomarker for health and disease. So we basically now understand what's going on. So again, to reiterate, the first step is you try to parcellate the brain into different regions of interest. The second is you kind of try to extract some kind of features like hippocampal volume or like the structural connectivity, like the white matter tracks. And the third step is you now build a structural connectome. And the way you do this is these red dots that you see, the red dots correspond to nodes, which is the region of interest that we're talking about. So it could be hippocampus and blah, blah, blah. The blue regions are basically what we get from the diffusion tensor imaging, which is the structural connectivity with two regions. And now we have kind of like a structural connectome. So again, this is a better visualization, I think, of the first step, anatomical parcellation. The second step, you do tractography, which is you try to visualize the tracks between different regions. And the third step is you now create some kind of matrix that measures how strong are the connections between two regions. So now we have talked about the structural connectome, and now let's talk about the functional connectome. Again, the first step of building a functional connectome is we need to define these regions of interest. So the same thing we talked about. The second step, which is specific to functional connectome, is we extract time series data from recording sites. What does this mean? Let's have a look. So remember when we say functional, we are recording brain activity. And time series essentially means we are recording brain activity for some period of time. And what that means is that the brain activity would fluctuate. It would change over time. And that kind of makes sense, right? Because every second we're thinking different things. So as you can see on the left figure here, it represents the brain activity of PCC, which is a brain area, or thalamus, which is another brain area, or thalamus for the subfigure B, and visual cortex in blue and green respectively. So as you can see, the activity kind of fluctuates over time. Now, how do we record brain activity in humans using MRI? So one of the ways is you can use something called bold fMRI. F stands for functional MRI because you can measure brain activity using MRI, and the functional stands for like the activity part. The bold stands for blood oxygenation level dependent or bold. And the way it works is, think about it. If you exercise and you're pumping your muscles, your muscle needs energy. The blood flows to your muscles, right? Because your muscles needs oxygenated blood. Same thing with the brain. If a particular brain area is active or is involved in some cognitive process, then that means that brain area needs oxygenated blood. When the brain needs oxygenated blood, there'll be a lot of oxygenated blood flow to that brain area, and you can use magnetic field to infer this flow of oxygen, just because oxygenated and deoxygenated blood have kind of different magnetic properties, and so by having a strong magnetic field, you can infer exactly how the blood is flowing to the brain. Let's try to interpret this figure. So the second objective of studying functional data is to often find whether two areas are functionally correlated with each other. So when the two areas are functionally correlated with each other, it means that maybe at this particular point in time, one area, like let's say at 30 seconds, PCC has very low activity, but thalamus have very high activity. So it's always opposite to each other. Or let's say for the subfigure B, you have like similar activities. So these are called functional connectivity because two areas somehow are functionally related to each other. So let's try for this question. If you look at the subfigure A, do you think the activities of PCC and thalamus are correlated or anti-correlated to each other? In other words, feel free to type in the chat. So in other words, do you think the activities of PCC, which is in red, and the thalamus, which is in purple, do you think they have a very similar brain activity? Or do you think when one of the brain area have a lot of activity, the other have low activity? So that's anti-correlated. Great, we have some answers, anti-correlated. That's great, that's awesome. So essentially now you know how to basically interpret the functional series, time series. Let's try another one. What about the thalamus and visual cortex? Do you think the activities are similar to each other? So when something goes up, it also goes up? Or do you think it's negatively or anti-correlated to each other? Like both blue and green of thalamus and visual cortex have completely opposite trend. Feel free to type in the chat. Yep, it's correlated. That's awesome. Thanks, Shahad. Great. Okay, so now we have that in mind. Let's try to interpret this figure. So just to recall, PCC and thalamus activities are anti or negatively correlated to each other. Thalamus and visual activities are positively correlated to each other. Now the question is, do you think the activity above describes which kind of sleep state? So is it SWS, which is slow wave sleep? Is it REM sleep or is it wake? Okay, and I know this is very hard, but I'm gonna try to unpack slowly and hopefully we can spend a few minutes on this. Okay, so if you see the subfigure A, it says region of interest, so ROI, correlations with PCC seed region. And on the y-axis, it says R. So R stands for correlation, right? So if you have 0.6, which means it's a positive correlation. If it's minus 0.6, it's anti-correlation. And the subfigure A, when it says PCC seed region, it means it's trying to see what's the correlation between PCC and all of the other brain regions. So what's the correlation between PCC and MPFC, PCC and DLPFC, PCC and visual cortex, and PCC and thalamus? We're talking about subfigure A, by the way. So if you look at the first bullet point here, we say PCC and thalamus activities are anti-negatively correlated, which means on the subfigure A, we only need to care about the PCC and the thalamus, right? The subfigure A. So which of the three bars do you think represent this negative correlation activity between PCC and thalamus for subfigure A? Do you think it's the black one, which is SWS? Do you think it's the light gray, which is RAM? Or do you think it's the dark gray, which is wake? Well, alternatively, you could also try for figure B. So what figure B is saying is, what's the correlation between thalamic or thalamus to all of the other brain regions? So we're talking about subfigure B. And if we look at what we know that thalamus and visual cortex activities are positively related. So for subfigure B, we only need to look at the last column, right? Which is the thalamic seed region. how does it correlate with the visual cortex? Which of the bars shows it's positive correlated between thalamus and visual cortex? REM, yes, it is REM. Great job. So as you can see, the REM is, yes. No, I understand. It's not easy. If you think about it, REM is the light gray bar, right? So for subfigure A, if you look at Arushi, your pointer to the thalamus, subfigure A, thalamus column. Yes, there, exactly. So you see that gray region, the light gray bar is showing that it's negatively correlated, right? I mean, the light gray bar is showing that PCC and thalamus are anti or negatively correlated because of the negative R value. As you can see, just by subfigure A alone, we can tell that the activity I'm describing here is basically showing the REM sleep. So anyways, if you have more questions about it, I'm happy to answer. This is the last thing I'm going to talk about for this macro scale, but essentially you can use this functional connectivity data to differentiate healthy people from someone with certain kind of disease. So an example is someone with Parkinson's disease would have a reduced visual functional connectivity. So you may be like, oh, visual stuff for Parkinson's disease? So as you can imagine that everything we do relates to vision, and therefore it kind of makes sense that for Parkinson's disease patients, the functional connectivity between visual areas, which is at the bottom. So as you can see, the visual areas CUN stands for cuneus, CAL stands for calcarine. So these are at the back of our brain, or in the figure is at the bottom. So these are responsible for basic visual processing. And you see, in healthy participants, is functionally connected to different areas of the brain, which includes things to do with memory, emotion, high order reward processing, decision making, speech comprehension, and so on. But in patients with Parkinson's disease, the functional connectivity between the visual regions to the all other regions like reward and emotion is reduced. And this kind of makes sense because actually if you see the literature, a lot of Parkinson's patients hallucinate things or they have some visual impairments. Or sometimes you can see this reduced functional connectivity even before you can see the clinical symptoms. So to repeat that, you can sometimes see the reduced visual functional connectivity in the brain even before you actually see an observable clinical symptoms. So anyways, that's just another way to see how connectomics really is relevant for health and also that vision is quite important. Okay, so now we have a complete picture of how to, extract time series from, let's say, fMRI, and now we can build a connectome, which is a functional connectivity or functional correlation between different regions. And yes, now I will let Arushi talk about the synapse-level connectomes.
So thank you very much, Sapu. That was very interesting. I will now take it from here and go to fly connectomics. So in this portion of the talk, we'll be talking about the impact of fly connectomics, and I will be illustrating this point through the work of our guest speakers. We hope that these guest speaker series inspire you to dig deeper into the different avenues of connectomics, and there for certain are many of those. And perhaps with one of these speakers, you might find topics that really resonate with you, and then you go deeper into it, and perhaps it might even shape the way your report is written. And the job for me in this talk is to allow you to get a glimpse into their work. So when you do go and dig deeper, you can, discover something really interesting. And when the talk comes, you can ask them really all your, I'm sure, wonderful questions or ideas you have, and, build a deeper appreciation for flight connectomics in general, and neuroscience. So yeah, one of the themes of our guest speakers talk is what insights has a synapse-level fly connectome offered? And to illustrate this point, I will do so through one of our guest speakers, who is Dr. Katie Schretter. Dr. Katie Schretter is a research scientist at Gerald Rubin's lab at HHMI Genelia, and she's giving a talk on November 5th, 2025, 9 a.m. ET. And her talk will be on understanding the neural circuits underlying aggression using the fruit fly. So a bit about Dr. Schretter is Dr. Schretter is interested in how the environment shapes social interactions and the underlying neural circuits. And her work has used to the use of connectomics, to the use of targeted specialized genetic tools that we have in the fruit fly, she has uncovered the neuromechanism that underlies female aggression. And I think her work is a beautiful example of how not just one, but many tools and the synergy of these tools together, the Connectome, the rich genetics toolkit that we have in Drosophila, and the neurophysiological capabilities we have together, we can uncover something really fascinating about the fly brain. And yeah, let's get into it. So her work is interested in how a fly's behavioral state affects its view of the world. So to start off, behavioral context has a strong influence on how an animals detect and interpret visual information. Let's unpack that for a bit. You see these two lions here, right? They look pretty aggressive. They're fighting. You can imagine they're not thinking about what's for tea. They're thinking about, I'm going to fight this lion, right? They're focused on their opponent. Or maybe something that's a bit more relatable is imagine you're driving in a really packed highway and it's just buzzing with a lot of cars. And again, the last thing you're thinking of is like, oh my God, I can't wait to sleep. It's more like, oh my goodness, I hope I don't crash with the person in front of me. And you're hyper focused. This is what fascinates Dr. Schretter and her colleagues. How do animals adjust our visual attention as our goals and environmental conditions change? And how does this look in the form of circuits? Because actually, there's very little known. So, Dr. Schretter and their collaborators wanted to understand how such changes in their focus or attention of an animal occur in the fly when the social context changes. For example, when a female fly enters an aggressive state. So, what does the social context aggression look like? So, aggression in the female fly would look like something like maybe she has some eggs laid in her territory. She would, if there's another fly coming in that territory, she's going to be angry because she wants to protect those eggs she has laid. Or maybe when she has a mate and another fly is a threat to that, she will be angry. She'll enter that aggressive state. And that social context is the aggressive state, and her visual attention changes. This similar thing you can see in vertebrates like starlings and meerkats. So, how did this project unfold? So, Dr. Schrader has been interested in female aggression for quite some time, and she's published a lot of things on this. So, her previous work found that ALPG neurons, which are in the central brain, they have nothing to do with vision, induce an aggressive state in female flies, and they found this through doing some connectomic analysis. So, what you can think of is ALPG neurons are like these aggression neurons. And then in this current project, or this paper, they went ahead and did some connectomic analysis, and something really interesting happened. So these are the ALPG neurons. These are these female aggression promoting neurons, and they connect to some downstream neurons. But what's interesting is that these same neurons are receiving inputs from these LC neurons. And if you guys are, fly compers and maybe have explored the optic lobe, you might recognize LC 9, 10, and 11 are some visual projection neurons. They're visual neurons. So that was pretty interesting. So, okay, you've got some aggression neurons and visual carrying neurons that are targeting the same neurons. So a natural question occurred for Dr. Schrader and her colleagues is, do LC neurons regulate the flow of visual information? And collective work in this field has also seen that the morphology and connectivity of neurons involved in arousal and aggressive states, such as ALPG, differs between the sexes. So in simple terms, ALPG neurons, aggression neurons, are in the female. And the counterpart of that in the male is P1 neurons. However, visual pathways are completely the same between the male and female. They don't differ. So this resulted in two questions Dr. Schrader and her colleagues had. Well, first is, with this shared downstream neurons they have, ALPG and LCs, do female aggression neurons or do female ALPG neurons modulate the flow of visual information? Is there some sort of interaction and crosstalk going on in here? And since ALPG is found in the female, but its counterpart is P1 in the male, but the visual circuits are completely the same between female and male, the another question is that, are there any similar mechanisms like this involved during other social interactions, such as courtship? Courtship is when a male pursues a female fly. And courtship is also a social situation where you would need to have visual information. You're tracking your potential mate. So these are the two questions that emerged from this preliminary analysis. And what Dr. Schrader and her colleagues found was that when the female fly enters an aggressive state, through these activation of these ALPG aggression neurons, this modifies the flow of visual information through 3 different mechanisms, which is pretty cool. So the first mechanism is a figure you're familiar with, is that ALPG, these aggression-promoting neurons, and these LCs, these visual information-carrying neurons, project to the same downstream neurons. And these downstream neurons connect to and modulate the activity of neurons that are to do with motor behavior. So this is one way that this aggression neuron, this social context that results in female aggression, is controlling that visual information, and it's controlling the behavior the fly has. Another way, another mechanism that occurs is through dendritic disinhibition. So you see these LC10 dendrites, these are normally inhibited by these lobular interneurons. A way you can think of it is like they're off. They can't do much. But what ALPG, again, this aggression promoting neuron can do is that it can relieve this inhibition, allowing LC10s, those visual neurons, to get more of an input. Again, this aggression neuron, this aggressive state in the social context is allowing for visual information to change depending on the social state this female fly is in. And the last one is a toggle switch mechanism. So I showed you that this dendritic disinhibition, ALPG can control the amount of visual information the female fly can take. But it's not even just visual information. There can be a switch mechanism where ALPG can decide which type of visual neuron it wants to its visual information it wants to amplify, LC10A or LC10C. As you can see from all these mechanisms that I've showed you, that this presence of these multiple circuit mechanisms allows the fly the freedom, flexibility to regulate attention towards various components of their visual environment. As you can imagine, there's a lot going in your visual environment. So having these different mechanisms allows different ways of when a female fly enters an aggressive state to control the amount of visual information she perceives. Now, onto one of the next themes about all things connectomics is how useful is a connectome? This talk, which we posted in fact today, I would like to highlight Dr. Srini Turaga's work, who is a group leader at HHMI Genelia, and their talk is this month, September 25, 10 a.m. ET, and they will be talking about computational neuroscience and AI and simulating the fly connectome. So let's get into what their work's about. Before I talk about their cool simulations and everything, let's talk about what makes a brain, or what allows for that neural activity. Well, I've been talking about synapse-level connectomes this whole time. One thing is those synapse to synapse anatomical connections that allow the brain to, have its neural activity. But another component is the dynamics of those individual neurons. Well, we now have a whole brain synapse-level connectome of the fruit fly. But remember, this fruit fly was a dead fly, and it was taken at one snapshot in time when it was dead. And although it's very, very rich in anatomical information, making equally detailed measurements of the functional properties, those dynamic properties, is something that's still not quite possible. So this ties into a question that people have been asking since the days of C. elegans. And many scientists have wondered, is a connectome even useful? I mean, it takes so much time to make one, so many resources, and after all, it's just a bunch of connections with its dynamical properties not really being there. And then they ask, well, now that we have one, how can we use the static connectome to predict how a neuron biologically responds under some task that it would in like a natural setting? And to do so, we kind of need to infer those computational dynamics of those individual neurons at synapses. Another way I like to think of is that these neurons, they have like a personality, like a characteristic to them, and which the connectome kind of doesn't tell you. It's just a bunch of connections, and connection A to B, there can be many paths taken. But which path is actually taken when a neuron biologically responds under a certain task? So that is a big problem. This is where Dr. Turaga and his collaborators work comes in. They had an idea. Why don't we deduce neural activity dynamics from our knowledge of the computation performed by the brain in response to certain inputs? Simply stated, Have you ever tried swatting a fly? I'm sure from personal experience, you know it's not a very easy thing. They are pretty damn good at escaping, right? Flies are really good at detecting motion. And in fact, if you've been playing with the fly connectome, you probably have heard of this motion path of the flies, and it's really, really well studied. And this is because this motion sensitivity comes because of the collective patterns of activity of networks of neurons and this dynamic properties each of these neurons have and the characteristics they have. So what Dr. Turaga's team and collaborators did was they made a neural network simulation of the fly visual system. But this neural network was connected the way a fly connectome was connected. However, like I mentioned, the static connectome doesn't really tells you all the information how they're connected, but it doesn't really tell you about how a single neuron kind of behaves, what's its character like, or single synapse. So they left those unknown. And this is where they use a tool in artificial intelligence known as deep learning to search for parameters that allow those neurons in the simulated network to detect visual motion like a fly would. So I will illustrate this point to this figure. So this is the neural network they made that constrained by the connectome. The connectivity is based on the connectome. And those single synapse and neuron parameters are unknown, so they use deep learning to search for parameters that allow this neural network to detect motion. And then they looked at the predictions of the resulting neural activity of this neural network. Similarly, because motion is studied for a very, very long time, very well characterized, they looked at studies from Drosophila where they measured their neural activity and they compared their predictions with the actual biological neural activity that we already know. What did they find? Well, how good is this neural network? They found that it's pretty good. So they compared their simulation predictions to experiment recordings of T4, T5, might ring a bell to some of you guys. Very popular direction selective neurons in the fly visual system. And these are studies encompassing more than 20 years, and there were 26 different studies. And what was really cool is that not only does this neural network predict which neurons of the fly visual system performed motion detection, T4, T5, but actually the mechanism at the single neuron resolution. So T4, T5 are two neurons that are direction selective. T4 responds to bright moving edges, and off T5A responds to dark moving edges. And basically, they have four subtypes which respond to different directions of movement. Essentially, if you actually go to the optic lobe connectome and if you see T4A, T4B, T4B, and C, whatever, you will see they actually end at different layers of the lobular plate, depending, and their orientation depends on the direction they are sensing. And it's pretty cool that the neural network simulation could actually capture that. What is the implication of this work? Well, one is that a lot of the fly visual system and generally recording from the fly is a really difficult thing. It's tiny. And that's why a lot of it is undiscovered. We actually don't know what most neurons do. And I'm sure when you actually found a bunch of neurons, a lot of them don't have much in the literature. So what this tool can help do is it's a great way to generate detailed hypotheses. So we can use a neural network simulation like this, and let's say there's a neuron, we don't really know what it does, and we see under a certain task, it behaves a certain way, and we can make a hypothesis of perhaps this is what it's doing, and then we can actually experimentally validate what we saw. So it's a very targeted way of testing what a neuron's doing. And another implication for this work goes back to the age-old question, how useful is a connectome? Remember, this neural network simulation used a connectome that was static, just anatomical connectivity. And remember, this connectome only has chemical synapse information, but even then, it's pretty surprising that it was able to predict in great detail the type of neurons that are used in motion detection. But imagine now, as you know, connectomes get better. We have information of neuromodulation, glial cells, electrical synapses, synaptic plasticity, and all included into it. Maybe this could be a really good tool to help predict what a neuron is doing under a certain task. So now that concludes two of the guest speakers that we've invited, and I hope that is as exciting to you as it is for us. So now I will be demoing a neuron example on Neuroglancer and neuPrint, and we will pick a very familiar looking neuron. If you saw Dr. Schretter's work, the LC10A visual neurons. So here is a gist of what your journey looks like, and we'll go one by one. So first, you need to go to Neuroglancer. So how do I go to Neuroglancer? So this is our website, and I don't know if Suppo's put it in the chat, but if you go to our website, everything's there. You can click on this thing here, and it should take you to Neuroglancer. Neuroglancer is a tool that is not working right now, but it helps you visualize neurons. And this is very odd. Okay, now it's working. Okay, perfect. So yeah, it helps you see neurons. So make sure you're on the right optic lobe version 1.1 dataset, because we are looking at the eye of the fly. Yeah, so first we click on ROIs. ROIs are region of interest, or in fly neuroscience terms, people normally call them neuropils. So we have 4 neuropils, main ones in the fly visual system: the lamina, the medulla, the lobula, and the lobular plate. So we can go ahead, click on ROIs, and those pop up. Should pop up. Hopefully pop up. Okay, while that's doing its thing, well, we should search our neuron of interest. So I decided to go with LC10A. So let's search LC10A. So if you click on one neuron, it should pop up. It seems like my internet's really bad. You know what? I think we'll have to shift gears a bit. But if you clicked on the ROI, this eye signal, this eye symbol here, you would have gotten something like this. You could have seen these layers. And really sorry, this is not loading, but we have made recordings of these in our previous workshops. So if you'd like a demo, we have it there. But for now, unfortunately, I'll have to just sort of have to use my PowerPoint slides. But yeah, you'll get some neuropils like this. Nothing's labeled, so it's a good idea to sort of memorize them or have something open. The way I remember a lobular plate looks like a plate, and next to the plate you have a lobula, and then you have the medulla and you have the lamina left. Yeah, and yeah, this is not working. So you have the ROIs, and your neuron will pop up, and you can see where its arbors are at. So this neuron will pop up somewhere like this. And if you click on the post-syn and pre-syn option, these options, you have to take the perspective of the neuron itself. So the pre-syn means this neuron, LC10A, is presynaptic to something. So presynaptic means it is these sites, these synaptic sites are outgoing to something. Whereas post-syn is that this neuron LC10 is postsynaptic to something, meaning this gets incoming connections. So yeah, this is really not working. So once you have your neuron, well, in this case, it's C2, but you can pretend it's LC10A. We will have to go into these things called since we deduced this, for instance, this C2 neuron is in the medulla here. So we'll click on the eye of the medulla, and you will get an option. If you click on something over at the side here, you'll get a bunch of layers. And basically with these bunch of layers, you can not only just see which layer, which neuropil your neuron is in, but you can see exactly which layer it's in. So you'll want to see where the arbors are in, and you can click on this. Again, it's a pity I can't really show this to you, but our previous recordings have this in proper detail. Then, once you've, which neuropils your neuron's going through, it's post-syn, pre-syn, like this pattern, it's all jumbled over the place, or it's pretty polarized, and you figure out, what layers it goes through, then we can go ahead and go to neuPrint. And neuPrint is a database that has connectomic data, and you can, use the web version to easily query details about this and analyze your neuron. Then when this pops up, you'll probably see there are a bunch of IDs when you write your neuron's name, C2, and this ID might show up. And basically, as Suppo mentioned, a cell type or neurotype, the cell type's the formal word, can have many neurons in it, so many cell types. So you just pick one, you click on the C version, it will give you this sundial thing here, and it can tell you the top inputs and outputs. So in this case, it's not very clear, but C2's top input is L5 and top output is L1 over here in yellow. So that's something you can also see. And you see inputs, upstream means inputs, downstream means outputs.
Would you like to try on neuPrint and show?
Oh yeah, maybe my neuPrint might work. Wow, today is not my day. Okay, LC10A. Okay, perfect. It works. Thank you. So yeah, so you see I typed LC10A. I got my neurotransmitter identity, which is acetylcholine. It's cholinergic, excitatory. This shows me the number of output connection, input connections it makes. And if you click on this, you can also see the names. But if you want like a really quick summary, you just click on the C. You can see, okay, this LC10A, top input T2A, top output LC14A1. And you know, by the size, these like the sizes and of these indicate how big of a deal it is for this neuron. So T2A, I mean, occupies a considerable amount of synapses on LC10A's end, and TMY21s lesser. So this is very important for it. Yeah, and this can also give you a brain region breakdown. So like a lot of its inputs are in the lobula, and AO2U is not in the optic lobe. And if you ever forget, you can always click brain regions. And if you go down, you'll see that AO2U is in the central brain. But yeah, gives you a lot of information, can do a lot of things. Next workshop, if technology is on my side, I will be showing you more advanced things that you can do on neuPrint. Okay, so now let's go to general feedback from submissions.
Just before you go over general feedback, I know some of you might be new and might be like, oh my God, this is very difficult. So if you go to our website, Marushi has kind of gone very quickly over it because we have covered it many times. However, if you go to the playlist, which is on the chat, you can see we have done multiple demonstrations of neuPrint. I suggest just look on any one of the workshop, it should be fine. Or you can also look at our guide, which again can be accessed in our website. And in our guide, we have it's like a manual, like step-by-step guide on how to use neuPrint. So I suggest you actually read that as well. And you should be able to do everything that Arshi has shown you today. But hopefully at least you have gotten a glimpse of how neuPrint and Neuroglancer works.
Yeah, and as always, if you have questions, you're more than welcome to reach out.
Yep.
Yeah. So general feedback from submission. So as you know, the early feedback was quite recently, and I've received a lot of incredible feedback. It's very exciting to see what you guys have been exploring. Yeah, and here's some general feedback which I think can apply to everyone for their reports. So cell type versus cells. I saw that there was a bit of a confusion between these things. I will show you how to do this in the next workshop, but essentially, or if you go to the supplementary figure of the Nern et al. Paper, you can also find the amount of cells, neurons in a cell type. Let's go with LC10A. There are 140 LC10As. So LC10A is a broad category. It is like the group, the name of the group of cells. There are 140 repetitions of this. And these are all the 140 LC10A neurons. And as you can see over here, these are all LC10A neurons. They're just different types. I did notice some people were confused and they thought like they're all a part of each other and they make an LC10A neuron. No, that's not the case. There are individual LC10A neurons. And yes, although they're in the same group, you might notice that when you go through individual LC10A neurons, their top inputs and outputs might be slightly different. It happens, but generally speaking, these are classified because of similar morphology, connectivity, neurotransmitter identity, and so on. But yeah, so that's one key thing to know. Cell type. Okay, so neuropils, layers, and neurotransmitters. Okay, so I noticed a lot of people skip on the layers part. This is really important, characterizing the morphology. So make sure when you're talking about your neuropil and talking about your neuron and which neuropil it's in, you make sure you actually see what layers it goes through. Its arbors are located in. And as Sapu I mentioned, we've extensively covered this in our other workshops if you need to ever refresh yourself. And something about neurotransmitter identity. And I thought this might be a bit confusing, so I want to clarify. So the neurotransmitter identity of neuron applies only to neurons it inputs into. So let's say LC10's inputs into MELO13. That's where LC10A's neurotransmitter identity matters because LC10A is sending its neurotransmitter to the dendrites or to the neuron MELO13, right? But when you look at the case of TM5Y is a main input to LC10A, LC10A's neurotransmitter doesn't really matter here because it's TM5Y that is putting its neurotransmitter to LC10A. So you see, in your analysis, even though you choose one neuron, it's very likely you'll have to slightly explore other neurons to see their neurotransmitter and get a better story of what's. Okay, so tips for figures and tables. So I was really happy to see a lot of people used a lot of figures and tables. Excellent. But always use a caption. Whenever you have a figure or an image, the judges will not know where you got the image from or what it means or why is it there. So it's really important to have a caption so it explains why is a figure important and why and how does it support your argument. And also the caption is not included in the word limit, so don't worry about it. Also, when you have figures, please include high quality figures. Judges need to see the details clearly and we want to look at what figure you've put in. Also, the use of technical jargon. So really, really scientific big words. Okay. You know, words we use in these workshops like arbors, I don't know, synapses, neuron. You should use them. They're appropriate to use there. But there's certain words which you might have found in your analysis through the literature. It's possible like the judges don't know them, and this is just good scientific writing practice. If it's not like a common thing understood, if it's not like something like a neurotransmitter synapse, it's a good idea to actually explain what it does and really see is it necessary to use that word. Also, clarity shows understanding. If you can explain really complex things in very easy language, that shows mastery, and this is a really important thing in research. So scientific writing, very important. And also for tables, please describe each table, the trends you see. Don't assume the reader knows anything. Actually, a really good caption would be, I read the caption and I blindfolded would know what the figure is about. A really good caption. So yeah, make sure you explain everything well and cite appropriately.
Actually, I wanted to add a few things on that. Yes. So when Arushi talked about high quality figures, and correct me if I'm wrong, there and always use a caption. I think Marushi is referring to two kind of figures. So first, obviously, is the figure that you take the screenshot from neuPrint. And I know that the text can be a bit small, so I guess like try to zoom in as much as you can and take a nice screenshot. That's one way. But another kind of figure that Arushi is talking about is when you're looking at the literature and you are screenshotting. So let's say you are seeing LC10 neuron and you screenshotted electrophysiology paper by Dr. Schretter. If she did electrophysiology, I'm not really sure, but I'm sure some of her papers have like this very complex looking electrophysiological recording. Now, imagine I'm not a neurophysiologist, right? Or even if I'm a neurophysiologist, I will not understand what those squiggly lines mean because it can just mean too many things. So and again, even if you include that very complex figure, you need to be able to point out why exactly you included the figure. So for instance, you might be like, look, this functional thing as shown in subplot A or B aligns with my XYZ connectomic analysis and therefore I conclude in a certain way. Like it has to be like that. If you just put a screenshot because you cited that paper, then you shouldn't include that figure because it's not helping your report, right? So just be clear about that. That including complex figures doesn't mean your report is good, but including figures that add to your argument and explaining it well, that's an excellent report. Sorry, feel free to continue.
Yeah, no, thank you. No, that was a very important clarification. Thank you. Yeah, just to be mindful of time. So I'll just move on to the next slide. So some more things. Um, when you do data analysis on neuPrint and Neuroglancer, check the literature. Does what you found match on there? I understand there are a lot of neurons where there's literally not much on, but if there is, you should compare and contrast from the literature. Also, no neuron is an island, and this goes back to my previous point some time ago, that like for neurotransmitters, it's likely you're going to have to see other things it's connecting to and see their morphology and the way they connect, and really dive deep in the analysis. Even though it might not be in your report, this is an important thing to have a more holistic understanding of your neuron because it's part of a network. It doesn't just stand alone. Also, be clear about evidence and hypotheses and all. If you propose a function or behavior, please make it clear whether this is a hypothesis you're making or this is something that you found from the literature. That needs to be very clear. Also, please support with the correct sources. Judges will be checking your references, and this is not to scare you or something. Like, try your best. I understand the literature is hard, but please use the literature that's appropriate for your work, not just randomly citing. We actually want to see if you understood what you read. And some of you guys skipped the reflection. This is a grading criterion. So if you forgot what that incorporates, please have a look at the handbook for more details. Yeah, and this is like an example of like some complex looking image that you might find. It's a good idea to explain or only screenshot the part that's relevant and explain like, why did you put it in? Why should I care? And what does this mean? Yeah, and I think this concludes our session. Yeah, let us know if you have any questions.
Yes, any questions? Meanwhile, sorry, just give me a second. I want to talk about Dr. Turaga's talk, just to let you know some opportunities that exist. Actually, I want to share my screen.
Okay.
So I suggest you, if you haven't signed up to our mailing list, because we actually send out a lot of emails, like something like this. So this is like an email campaign, as you can see here. So we send like a bunch of emails out and we explain, we give some kind of tips and tricks to read his work. So we're like, okay, you're welcome to check that group's incredible work, his recent paper here, which is what Arushi has explained today. And how do you register? So you click on that link and then you type your name and so on. And I also want to share an opportunity that was shared through the email and through Discord. Essentially, we want to facilitate discussion of Dr. Turaga's work. And so, to this end, we have created a Discord thread here, which I'm clicking. But first, you kind of need to join the Clematis Discord first. As you see here in this thread, we ask students if you want, this is optional. We encourage you to share creative work that discusses Dr. Turaga's work and post it on that thread before September 20th on the Discord thread. And this could be like a short video, an infographic, or any other form of creative submission. And if you recall in our deliverables, you have to do some kind of creative submissions anyway. So I think this is kind of like a nice practice. And you can also go look at our Discord to look at how other people have been submitting. People have been creating such wonderful and creative videos. And I hope you can be inspired by some of those submissions. And many of them are high school students as well. So the idea is you do some kind of creative piece on Dr. Turaga's work, and if we find a submission that's good, we'll be spotlighting essentially the best submissions during Dr. Turaga's talk. So I think it's a way to put yourself out there and we will select your work. And I think if I was an undergrad or high school student, I would really want for Dr. Turaga to see kind of my short video infographic on his work. And I think that would be pretty amazing. And yeah, so I wanted to share about that and a quick run through on, so as you can see the website, you can see, I suggest you join our mailing list. You click here and then you can write your emails. You can also click this handbook. So in a handbook, there are four. Essentially, the first one is the actual handbook, and we have like selection criteria and everything. The second one is the starter's guide. Oh, someone is drawing on the thing. So here is a starter guide. So we try to explain like very basic neuroscience and the optic lobe, the data set, and so on. And we also have kind of tutorial, which Archie very quickly went over it. The third is the research report template, and hopefully you can use this as a guide. And the last one, also quite important, is you might be like, what do you need to know in order to succeed in this competition? So we really try to map essentially the knowledge you need to know to foundational undergraduate and high school curriculums. Of course, some things will not be taught in high school curriculum per se, and I think that's the point of competition. But essentially, a lot of things that you'll be doing builds on what you would be learning in high school and undergrad anyway. So for instance, like what's a neuron, what's neurotransmitter, pre-synapse, post-synapse, something like how behavior, cognition, and connectomics kind of link together, or like how to use Neuroglancer and neuPrint. This is very specific to Synapse-Level Connectome. But essentially we have like point by point to explain what you need to know to write a nice report, I feel. So anyways, that's it from me. I hope that helps for those who are new. Do you think I should show very quickly the Neuroglancer that you missed? Because my Neuroglancer seems to be working.
Yeah, you can do that.
I
Yeah.
could. And maybe Arushi, you could guide me, because again, I'm not an expert in fly connectomics. So that's the data set. And you click ROI, and then slowly you see these pills. So let's quiz some people. What is this middle layer here? What do you think is this middle layer, this big layer? What kind of neuropill it is? To give you a hint, I'm gonna go to this guide. Yep. So what's the middle layer? The middle layer is the medulla. It's ME. And so these are the region of interest. And let's try LC, what is it, Arushi? LC10, right?
Yeah. A.
And as
So
Arushi would always have mentioned in previous workshop is you make sure you capitalize the letters and make sure, so let's say if you do like LC, you see no match. So you need to make sure you write it's case sensitive essentially. So let's click on one of the random neurons. Let's try a pink color neuron. You see these are neuron types, LC10A, because, and remember LC10A, although it's a cell type, it can have many LC10A, right? And they are indicated by different IDs. Same thing like having 10 green lays in a department store, even though the green lays are essentially the same type. Okay, so we have a neuron. And this is the soma, is the cell body.
mhm.
And the soma obviously is the center of the neuron of the cell. Then what are we supposed to cover? Pre and post synapse, right?
Mhm.
So the way you see where in this neuron does it receive information or does it send out information, you click pre-syn and post-syn. They're basically the synaptic sites. So as you can see, post-syn is in blue and pre-syn is in red. It's not too clear here.
Mhm.
But if you notice, this is a bit different in human neurons in that the area where it receives and sends out information is pretty much intermingled with each other. And that is why you can't really say this is dendrites or this is an axon, just because, or axon terminal, just because all of this, like this very same area, both receives and send output information. So if you notice, Arushi is very careful with the words she used. So we call this, is it an arbor? Is that the right term? Sorry, I can't see you. So you
Oh
have to,
yeah, yeah, sorry, it is.
yes, it's an arbor. And arbor is essentially tree-like branches. Now, what was the next thing on the agenda that you're supposed to cover?
So which neuropil is this in?
Which neuropil is this in? Okay, so let's see. Let's test my fly anatomy. Wait, how do I? Okay, so you basically press the finger and you do like that.
Yeah, yeah, yeah,
That's how
yeah.
you rotate the brain.
Hmm.
Right. So this is the lobula, is it?
Correct.
Right? So remember at the bottom, it's either the lobula and the lobular plate. Archie says lobular plate looks like a plate, but sometimes for me, I just think it's purple color. The color doesn't change, so that's the lobular plate. That helps too. And the big thing is the medulla.
Or medulla in the middle.
Yeah, that's a good way to memorize. It's the medulla in the middle.
Yeah.
Okay, so is that
Now, you know it's in the lobula. How do I know which layers its arbors are in?
right? So although, as you see, this neuron kind of goes everywhere, but what we care about is where in the brain area does it integrate information, which is it receives or it sends information, or in other words, where the synaptic sites are. So we know it's in the lobula, but which layers? And Arshi, can you explain what to our audience what layers are?
Yeah, so layers are something that are kind of back in time in the days of when they would before the connectome, people had to stain these neurons to actually see their anatomical features. And to classify these neurons, to classify them into cell types, layers were introduced because you're maybe thinking, how do you know if a neuron is an LC10A and how is it a C3? So one of the ways is the way it looks, right? Maybe they look a bit different, but that's not enough because at the end of the day, they kind of all look like a crazy mess. So one of the ways is to see where their arbors are in which layers. If they, start at layer 1 of this neuropillar and at layer 4 of that neuropil. So that's the reason they're there and exist. So that's why they help with actually morphology.
Okay, we have a question. Is it possible to integrate this work into my school research assignment? Absolutely. If your school requires you to, I don't know, produce a poster, I know a lot of people might do like a literature review, but you can do literature review, but you can also just analyze the real data. Like, isn't that exciting? And you can combine the connectomic analysis with the literature, and you can maybe even propose something new. So definitely, and remember, school research assignment can be different levels, right? And actually, we have this website for educators, but students can have a look at it too. But essentially, we try really hard to map to the high school curriculums and also undergrad too. But essentially, if you are a high school student, which I'm assuming here, like for instance, I don't know if any of you are doing IB. I did IB biology, so you can see that this competition was designed to help if you are learning about chemical signaling, neuro signaling, and integration of body systems. So these are like the syllabus points in the IB biology curriculum. You can actually use this competition to fulfill the internal assessment, which requires you to, for instance, work with some kind of research. Either you do an experiment or you do kind of computation analysis. So our thing will fall under extraction analysis of data from a database. But it can be from AP curriculum. It can be any kind of school-based research requirement depending on where you are in the country. So absolutely. And if you have any questions on how you can do that, you can always get in touch with both of us on Discord or our email, and all of the contacts in our website. Thank you for that great question. And I hope that helps for folks who might be interested in actually using the neuroscience data for some of your research requirements. Anyways, so now I guess the goal is to look at how do we describe where this particular neuron is in the medulla. And one of the terminology we use is this is in XYZ layers. So Arushi, can you explain step by step how we can do this? Our
So you
layers
know the the arbors of this neuron are in the lobula,
here.
right? We've identified that. Now I would go to LO. LO stands for lobula, and I'll go to layers.
So I click here, right, on the I
Yes,
button.
I, and then you
Great.
click, and then you go back to LOR layers. Go back, hover over it, yeah, and you see, yeah, you click on that.
Right. So you click this, I guess it's like a filter kind of button.
Yeah, and you can click on the big eye at the top, like
Great.
yeah,
So
this one.
here the lobula have 7 layers. Is that correct?
Yeah,
Right. So these are the 7 colors. So let's click the eye, which will show all of the layers. Give it some time. Right. Here we go.
and one by one we peel like an onion, and we find out where its arbors are concentrated in which
Right.
layer.
So can you see this last one on the left here is layer 7, as you can see, like this gray thing also. When Arashi says peeling one by one, so we want to slowly peel these layers to see can we observe the synaptic site. So let's peel layer 7 off. So can you see maybe very little, but I guess very few is in the layer 7, but not really, right?
Yeah,
Let's try and peel some more. Let's see. Oh, there we
yeah.
go. So a lot of it is in layer 6. As you can tell, because if we cover it with layer 6, you can't see any synaptic sites. So now you can say for sure that well, I assume, oh, so I guess it's just the postsynaptic sites that's in layer 6. That's interesting. So let's do this. So now let's peel off layer 5. Great. So of course, so it seems like the sites is in layer 5, but not the presynaptic sites. Really? Okay. So let's go to layer 4. Wow, so it seems like the synaptic sites are covering layer 6, 5, 4, I guess 3 as well. Yeah, I think it's 3.
So I would put 3 back on,
You put 3 back on.
and then remove 2 and 1 to see from the other side.
Okay,
A bit easier. I would remove 1 first.
right. So you actually cannot see anything at layers 2 and 1. But of course, so now we can tell that these synaptic sites are at layer 3, 4, 5, 6, like mostly. So that's how you can use to describe the neuron morphology of LC10A. That's great. So thank you. Is that all we were supposed to cover for
Yeah,
now?
that was all.
Excellent. So thank you all for staying on for our next workshop on September 9th. We will be focusing on the more computational techniques in neuropsychiatry and also more nuanced and advanced technique that you can do on neuPrint. So thank you, everyone. Have a good day. Bye-bye. Thank you.
Thank you.
Am I going to see you guys both again on September 9th?
Yes, we are giving the talk. We will be mostly giving workshops. Like if you see the infographics, it will have both our names on. That means both of us will be there. If we invite the guest speakers, then both of us will be there as well, but just that the guest speaker will be giving a talk instead of us. Yep. So
Oh, I'm glad to hear that.
great. Okay. Looking forward to seeing you again. Bye-bye.
Thank you very much ค่ะ See ya สวัสดีค่ะ Thank you very much ค่ะ
Bye-bye.
Clematis Research Empowerment Hub. Empowering research for everyone.